So, hi everyone, this is our final presentation. We're creating a polymeric heart valve replacement for TAVR valves. My name is Jan.
I'm Matea.
I'm Kyla.
I'm Michelle.
So, one of the leading causes of death in the world is heart disease. 300,000 Americans suffer from aortic stenosis, which is valve narrowing every year, and 106,000 Americans need heart valve replacements by the end of the year. There's two major reasons for this. One is regurgitation of blood into the left ventricle again. Two is stenosis, which is valve narrowing caused over time through calcification. There are three major types of heart valves available on the market: mechanical, bioprosthetic, and TAVR. The mechanical have a long lifespan but have risk of blood clotting. Bioprosthetic have risk of calcification, no clotting, and a shorter lifespan. And TAVR is great for patients at risk of complications during open heart surgery because it's minimally invasive. There's clinical trials being done right now for tissue engineered heart valves, which have the ability to regenerate because they're made of biologically engineered matrix. And competitors for all of these heart valves are Boston Scientific, Edwards Life Sciences, and so on.
The design of our TAVR must meet specific objectives and requirements to ensure long-term safety and functionality in the patient and address drawbacks that are not entirely met with current TAVRs. So we are considering design criteria encompassing hemodynamics, crimping, and stresses that are applied in the body during the cardiac cycle. The specifications listed were created to reflect a properly functioning heart valve replacement. Crimping functionality is very important for our design because the device must crimp stably while preventing crimping damage to the surrounding collagen fibers, which is an issue that has not been addressed by current TAVRs. A crimping force of 82 newtons would address this issue based on research for compatibility testing in the aorta. Furthermore, based on values from radial force profile testing and computer-based device thermogenicity emulation, or DTE, specified values of crimping force, bending and shear stresses, and regurgitant flow are necessary for safe and effective polymeric TAVRs for desirable outcomes like stability, non-calcification, and anti-stenosis.
We designed a 3-leaflet heart valve made from the polymer X-Sibs. The leaflets of the valve will have a thin cobalt chromium mesh embedded within and a cobalt chromium stent for balloon inflatable deployment. The leaflets will be convex and angled in the direction of blood flow to help minimize shear stresses that often lead to thrombosis and the effects of blood pressure. The polymeric material will have a 1% non-eluting heparin coating. The stress on the stent will be 80 to 90 micrometers to maintain strength and stability.
So our advice has advantages and limitations. To begin with the limitations, there's a high load during crimping. The concave valve shape makes it more difficult to close due to the blood pressure on the valve. And then there's potential for paravalvular leakage due to this. Some advantages are that the concave leaflets minimize radial forces, which is good. There's flexible polymer leaflets which provide the strength that a mechanical valve leaflet would with the compatibility of a bioprosthetic valve. And then they're great for younger patients in need of valve replacements because it's minimally invasive, and the heparin coating allows for no blood clotting to form.
For the production of our design, we focused on the criteria for each component, including the stent material and deployment, the heparin coating, the leaflet wire mesh, and the X-Sibs polymer.
Medtronic performed several tests comparing stainless steel and cobalt alloy stents and found that cobalt alloy stents maintained a strong biocompatibility but have a higher ultimate tensile strength, as seen from the stress strain curve to the right, showing that they can withstand the stresses placed on the stents during blood flow. Medtronic also found that cobalt chromium has a higher density than stainless steel, which allows for very thin stent struts while maintaining strength. Thin stent struts provide many advantages, including radial strength, reduced risk of recoil and restenosis, and resistance to structural damage during crimping. The cobalt chromium stent proposed by Medtronic was also found to conform to the ASTM F562 standards, which are for a cobalt alloy surgical implant.
So our design will incorporate the balloon expandable deployment mechanism. So we looked at stresses produced during crimping of the valve. The image on the bottom right compares variation in von Mises stresses throughout a cobalt chromium stent during crimping with maximum stress throughout the stent being 750 MPa, in which the valve will need to withstand. We also looked at the interaction between the stent and the surrounding aortic tissue during deployment of the valve. During insertion, the crimped valve is expanded by radial forces through the balloon expandable mechanism. And so one study analyzed the mechanical properties of the Medtronic core valve and found these radial forces to be between 44 and 69 newtons, which translates to hoop forces of about 7 to 11 newtons. And as a result of these forces needed to expand the stent, the surrounding endothelium tissue triggers an inflammatory response. Initially, upon deployment, there's little to no smooth muscle cell proliferation around the stent, and instead, thrombocytes surround the struts of the stent, pushing against the heart walls. So, part of the reason for using a thin strutted stent is to reduce friction at the tissue stent interface to reduce thrombocyte formation as an initial inflammatory response. And then during thrombocyte coagulation, lymphocytes migrate to control and regulate the corresponding inflammation. And 2 to 3 weeks after implantation, an increase in SMC proliferation occurs, which is followed by SMC migration to the endothelial lining of the walls to engulf the struts of the stent. And then during migration, an extracellular matrix is formed along the endothelial walls, and the interaction between this ECM and the SMCs form a thin lining that interacts with the stent struts.
The leaflets of the valve will contain the polymer X-Sibs as it has favorable mechanical properties and can decrease stress concentrations on the leaflets shown in previous studies. X-Sibs has nonlinear hyperelastic behavior in that it can undergo over a 100% strain and still fully recover. It also will be able to withstand the maximum stresses seen across the leaflet, as it has a strength of about 5 megapascals. It also has been seen to have similar interaction with blood as normal tissue valves. In one study, as seen in the graph on the right, based on hydrodynamics testing using DTE, it did not appear to cause a significant increase in platelet activation that would cause thrombosis when compared to regular tissue valves. Our device has a cobalt chromium mesh embedded within the polymer to enhance the durability. As discussed earlier, cobalt chromium can be made very thin and retain its strength. In a study that compared several different metal stents, the stent made from cobalt chromium had one of the highest flexibilities and moderate strength. The strength of the mesh can withstand the bending and shear forces, and the very thin nature of both the metal mesh and polymer will ideally allow for these stresses to be minimized and in turn improve durability. The risk of thrombosis is reduced by placing the mesh within the polymer, so there is no direct metal blood interaction. However, after adhesion bonding of the metal and polymer, the constant stresses placed on the leaflets may cause friction between the two materials and ultimately degradation of the polymer.
So on the left, you can see we considered two ways to incorporate a heparin coating onto the leaflet surfaces, either as an eluting or non-eluting coating. One study compared ionically and covalently bonded heparin coatings on a PET polymer. While the results showed a higher activated partial thromboplastin time for the ionic coating, researchers found increased bioactivity and reduced platelet adsorption in the covalently immobilized coating compared to the ionic coating. So this is why we chose to covalently immobilize the heparin to the polymer, which can be done in a variety of ways. On the right side, we're showing the functionalization of PPO to bind to heparin through the addition of PO spacers conjugated to the styrene portions, which could be translated to the X-Sibs polymer. And then two other studies were used to evaluate the effectiveness of incorporating heparin onto our device. The study on the left characterized the binding surface energy of a covalently immobilized heparin coating on a replacement aortic heart valve, which was found to be 169.5 electron volts. And then the study on the right compared thromboresistance in ePTFE grafts with the covalently bonded heparin coating. The coated graft resulted in a 95% thrombus-free surface and a heparin coating or activity of 24.7 picomoles per square centimeter, which are two target values our device would aim to exceed in order to maximize antithrombin binding to reduce thrombus formation. So our device we could conduct experimental assays to compare antithrombin uptake to different heparin concentrations. which based on the concentrations used in these studies should be some concentration between 1 and 35 micrograms per square centimeter.
There are international standards and regulations that are applicable to the TAVR. ISO 5840 Part 3 is applicable specifically for transcatheter heart valves and delineates the methods of manufacturing it and the verification and validation tests of the design. Design qualification would measure the physical, chemical, biological, and mechanical properties of our TAVR, and it would also define operational and performance requirements. On the other hand, the international standard ASTM F2477 would also be applicable for our TAVR. It would outline the determination of durability of the TAVR stent, and test methods would be applicable for the cobalt chromium stent. F3211 Part 17 provides a standard guide for fatigue to failure methodology for cardiovascular devices. So for our TAVR design, it would aid in providing the use of fatigue models and statistical techniques for estimating these mechanical properties. And it would also provide the framework for assessing the TAVR's longevity. And in terms of sterilization, to consider sterilization techniques, ethylene oxide sterilization would be the most optimal. So according to the FDA guidelines, ethylene oxide sterilization would be compatible with moisture or heat-sensitive polymers and polymers with hydrophilic coatings, since the device would not be prone to any structural valve deterioration. It is optimal for polymeric devices like ours because it would not deteriorate the device during the sterilization process. To ensure safety of our heart valve replacement after sterilization, it would follow the guidelines outlined in ISO 11135. The catheter would also be prepared for sterilization by saline flushing and submerging it in a cold saline bath.
Mechanical and in vitro testing of the device follows the ISO 5840 Part 3 guidelines. The following tests will be done on the largest and smallest valve sizes. The samples should be tested after crimping and deployment. Several mechanical tests, including tensile, compressive, and bending tests, should be performed on the leaflets to find the stiffness, modulus, and strength. Durability testing should be done according to the ISO guidelines using a 400 million cycle fatigue test. The failure pressure of the valve could be found by pressurizing the leaflets in the closed phase during a back pressure failure test. Hydrodynamic testing could be done using a pulse duplicator to see how the replacement valve behaves in an environment similar to within the body. The strain of the leaflet during flow could also be seen using high-speed imaging, as performed in several other studies.
For biocompatibility testing, the team will be following ISO standards 10993. And so with that in vitro testing that we plan on doing, we plan on finding the hemocompatibility of the device, which assesses platelet adhesion and thrombus formation through scanning electron microscopy. We also plan to test a flow of blood through the valve to see if there's any damage to the blood cells. Additionally, we are planning on testing for the degree of immobilization for heparin, which Michelle spoke about a little earlier, to see if it's the right concentration of heparin on the device using a blue dye assay. We plan on doing cytotoxicity testing, which we'd be culturing other types of cells on the valve to see if they're viable. And so we plan on sterilizing the device, as Kyla had mentioned. For in vivo testing, we plan on using an animal model. We chose the animal model of a sheep because it's recommended due to its similar blood pressure and heart rate values to a human. So for our animal trial, we'll be following ISO standards 5840. First of all, the study must be at least 90 days, but the team plans on testing for a little longer to ensure proper results. Our control will be a sheep implanted with a TAVR valve currently available on the market, so we can compare our product with the competitors. We plan to assess thrombosis on the valve through post-mortem examinations performed on the test subjects. We plan on testing for calcification and mineralization by regularly taking photos or radiographic documentation of the heart valve inside of the test subjects. And then animal blood pressure and vital signs will be measured regularly and compared to their baseline values in order to ensure proper functioning of the heart. We plan on testing with 6 to 12 sheep in order to ensure we have a proper sample size.
Thank you.